Finish up.
Oh, they are? Okay, we're just waiting for them to stop talking.
Alright. So let's talk a little bit about how these microorganisms can cause infection, which is myself. Have we talked about the difference between endemic, epidemic, pandemic, and
No.
because some of this stuff we're going to talk about and don't remember what I'm talking about.
We did a little bit before.
Me either. Okay. So we talk about endemics. So endemics are infectious diseases you find in a certain amount every year, and we expect to see that number of cases. An example of this, maybe an over the top example, I guess, is out in the western United States, prairie dogs carry Yersinia pestis, that's what causes the plague, the bubonic plague. And every year you see about 120 to 180 cases of that in the southeastern United States or southwestern United States. What happens when the prairie dogs have fleas on them? Fleas can transmit plague, and what happens is a house cat or something goes and tries to get a prairie dog, and he comes back and gets fleas on them, and then they give the fleas to their owner. Or sometimes what has happened is they actually kill the prairie dog and leave the prairie dog on the owner's doorstep like a little gift with fleas all over it, they're carrying plague. Or sometimes you get somebody that decides to go try to play with a prairie dog that has fleas on it that's carrying plague, and they get it directly that way. But there's about 150-ish cases of that every year. So that's endemic. We expect that. That's kind of what happens. Now, if that rate increased to where it was 1 year it was 200 or 300 or 400, at that point is considered an epidemic. So it's above that normal kind of baseline. So that's an epidemic. When you have an epidemic that's global, so like when we did, like when we went through COVID-19 and all that stuff, that's a pandemic. So pandemic is just a global epidemic. Different examples of pandemics: COVID-19, so SARS-CoV caused by SARS-CoV-2, SARS-CoV-1, which we'll talk about here a little bit later on. The 1918 flu, which is called Spanish influenza, great influenza, so that was a pandemic. You see occurrences of different, not as much anymore, but in history you see different epidemics of cholera and influenza and tuberculosis and things like that. Everybody knows what chikungunya is, right? Yeah.
I just have a question. So you know how the common there's a rise of measles going on in the US? Would that be epidemic or endemic?
So if it's in the United States, it would still be considered an epidemic. Now, if that's something that's spread globally, like due to air traffic, usually, then it becomes pandemic.
So this is stuff in one state, it's just endemic.
If it's in one state or one part of the planet, it's considered epidemic. As soon as it's everywhere, like globally, like SARS-CoV-2 was, then it's pandemic. Yeah. When you think pandemic, think global. Epidemic is just certain area. Chikungunya is transmitted by mosquitoes. So what it can do is it can cause shrinking of the head in infants. If you have a newborn, it can cause some neurological defects or potentially death. And an adult, it causes things like fever, joint pain, things like that. Polio has caused pandemics in the past, and a bunch of other things, plague. There's a bunch of different kinds of infectious diseases that cause things called typhus. All that really means it's a very wide term. Anytime you see the word typhus in the general sense, usually what it means is it's some kind of infectious disease that was spread by a flea, a tick, or, flea, lice, chiggers, things like that, and it causes fever. That's pretty much, which is a lot of different things. But that's what typhus in the broad sense means. There's a lot of different diseases like that. A difference between communicable versus non-communicable. So communicable means it's communicable, which means you can transmit it from one person to another. Non-communicable means it's not communicable, which means you can't transmit it from one person to another. So there are infectious disease examples of both of these, and then there are more obvious examples of these. So communicable disease, influenza. Somebody comes in here, they have the flu, they sneeze, somebody else breathes it in, they sneeze in their hand, they touch a doorknob, somebody else touches the doorknob, they rub their nose, things like that. That's a communicable disease. Non-communicable, maybe you have Helicobacter pylori infection. We talked about that causes stomach ulcers. It's not really communicable from one person to another. So there's some infectious diseases you can't spread from one person to another. There's also, again, non-infectious disease examples of this. Heart disease. It's not communicable. Cancer, but well, maybe I shouldn't use that example. Because there's different things that cause cancer. So like HPV, you can spread from one person to another, and that could, for the most part, cancer is non-communicable. Heart disease, high blood pressure, high cholesterol, those are things, disease processes that you can't transmit from one person to another. And then there are vector-borne illnesses. So those are transmitted through an insect using an arthropod. So what's that mean? Things like mosquitoes, ticks, sometimes flies, things like that bite you. The infectious disease, the organism that causes infectious disease, part of their life cycle is in that insect, inside that vector, and they need that vector for part of their life cycle. If they don't pass through that vector, then they don't live. So they need it. And what we usually are is like an accidental or an incidental host, which means that flea or tick or mosquito or whatever bites us and then transmits that infectious agent to us. We're not necessarily part of that life cycle, we're an accidental host, like a OC. Yeah.
One question. It's a little bit loaded, but when I had a class with Dr. Lee, she always kind of made a motion to say that if you don't use it, you lose it. And I was looking for like your immune system, and like if you don't get sick occasionally, like your immune system can, I don't know, it's bones, so to speak.
If you don't have immune stimulation,
So then you get like the whole constantly like sleep again and don't, you know, just take precautions all the time. Maybe lose that.
so immune stimulation is a good thing. So your normal flora, even in your gut, helps stimulate an immunological response. We talked about that a little last time, didn't we? Yeah, we did. So before age 4 and 5, there's stimulation for things like that as normal flora. If you don't have that, then those people, yes, get a lot more sicker. If you use a lot of hand sanitizers, I think you put that in your, like old school Purell and stuff. So like old school Purell is pretty much rubbing alcohol. There wasn't really anything else in it, which kills things pretty well. The only thing is what's alcohol do when you put it on your skin over and over again? That just starts to dry your skin out. Well, then what happened to those people? And they're like, oh, these people are getting like little micro fissures and stuff in their skin. Well, then you get infections in that because you've broken like your skin barrier. So that's when they started adding aloe and things into it to moisturize as you do it because they're like, oh, that's bad. So that's kind of not necessarily an immunological answer, but I mean, sometimes those different disinfectants and things can push resistances. I think I talked about this in microban. Like microban, you used to put freaking everything that was a plastic. It would leach out over time. But what it does is that actually selects for bacteria. They're like picking up every plasma they can find, and what's carried on plasma is resistance to that. So you start to select for bacteria that are resistant to literally everything. So that's why you don't see microban effects anymore. So it's not necessarily like with a lot of those compounds, it's not that it's or disinfectants and things like that, antimicrobials, it's not necessarily that it's an immunological thing. It's that there's other things that they can do to cause damage to like your skin, or they can select for plasmid uptake or things like that. The more things you're exposed to, the better. But did I answer that at all? Okay, so there are epidemic curves depending on what kind of a disease process you're talking about. So what's that mean? So over there with the red curve, see this is letter A here, almost said number A. Wow. Common source epidemic follows a sharp spike, then quickly falls over time. So an easy example of this to understand is something that's associated with food. So in my path microplastics, I think give an example of like peanut butter. I think there's a salmonella outbreak of peanut butter. So number of cases going up. There's salmonella in peanut butter, everybody's eating peanut butter, and then, oh, There's some done peanut butter. They have a recall, the cases drop off because it's a common source, the peanut butter. So once the peanut butter is taken off the market, it goes away, you don't see the cases anymore because people can't get salmonella from the peanut butter because they can't buy the peanut butter anymore. So that's a common source. So food poisoning, anthrax, depending on how you come into contact with them, that can be a common source, whether that's a bioterrorism event or like if you're talking about somebody, they're on a farm, they're tanning hides and making leather, maybe the cows got that from the soil, and there's people that have all been working with that livestock and now exposed to it. It's a common source. Once the livestock, no more common source. And what they do with those livestock is if they know that they're carrying things like anthrax, they call the herd and they send them to the Department of Health, usually from the state, to do necropsies on them. We'll talk about this. That is Legionnaire's disease. We talked about that before too. You have a fountain or something where there's Legionnaire's disease in it. People are exposed to the fountain. Common source. They get Legionnaire's disease. Ran the fountain, no water in it, no water, no Legionnaires. People have Legionnaire's disease. So common sources. So common source usually is talking about a non-communicable disease. Usually. A propagated epidemic is usually talking about a communicable disease that can be transmitted from one person to another. And it being transmitted from one person to another is how it propagates. So a propagated epidemic follows a gradual rise in declining cases over time. So you see that represented with B, and that's indicative of person-to-person contact. So flu, COVID-19, things like that. So if it's more flattened, it's probably propagated. If it's more spiked, it's probably common source. Pathogenicity, so some definitions here. Pathogenicity is the ability to cause an illness or disease. Pathogenicity is the ability to be a pathogen. Does it make people sick if they come into contact with it or not? So pathogenicity is, does it make you sick or not? Virulence is how sick does it make you? So if you're talking about virulence, it's already, you know it's a pathogen. It's just how sick does it make you? So it's a measure of the pathogen's ability to overcome host defenses. The virulence is usually determined by whatever arsenal of virulence factors the pathogen has. Does it have hemolysis? Does it have coagulases and collagenases? What does it make? Does it make LPS, like endotoxin? So those are all things that impact if it's how sick you get from it. If you have a bacterium that makes 83 different virulence factors versus a bacterium that only makes one really weak virulence factor or no virulence factors at all, the one that's making a lot more stuff is probably going to make you more ill, generally. So that arsenal of virulence factors is determined by what genes the bacterium has. So those genes can be on different pieces of parts of the genetic makeup of the microorganism. It could be on the chromosome. It could be on plasmids. Remember, plasmids can transfer from one organism to another. They could be on mobile cassettes, pathogenicity ions, and transposons, those jumping genes. Some bacteria actually have different toxins encoded as part of the phage. So there's a bunch of different places they can be. And what happens is when you have a bunch of these different pathogens in the same environment, like a hospital, because a bunch of people come in sick with different illnesses in the hospital, you have a lot of nasties in the same place, they can swap that genetic material between each other. And that we talked about promiscuous versus non-promiscuous, right? So there's some things that can be transferred just between like E. coli to E. coli. And there's other things that we can transfer pretty much between anything. Coli to salmonella to Vibrio to Klebsiella to whatever. Those are the ones we really worry about because they can transmit those resistance genes between one another. So that's why you sometimes see some really nasty drug resistance in the hospital. The virulence can increase when pathogens faced with different environmental stressors. I think I talked about this a little bit here on one of these days too. Is sometimes what happens is if there's a stress, an environmental stress that the bacterium is exposed to, it actually triggers a stress response in that bacterium, which makes them upregulate different virulence genes, which means that they start getting a lot angrier and they can make you a lot sicker. I don't think maybe I can see the same thing here. But one of the organisms I work with, if you expose it to a little bit of ethanol, so not like 7%, like 1 or 2%, it actually induces it to make really thick biofilms. So it elicits a stress response. That stress response upregulates the ability of that microorganism to make biofilms and things like that. again, so pathogens can make virulence factors. Virulence factors are actually what's going to make you sick. I talked about this, I think, a little bit before, but maybe not as much detail as I'm about to here. The way you measure virulence is by looking at things like the ID50 and the LD50. So the infectious dose 50, say you're doing mouse experiments. For a lot of these experiments, they actually use ferrets, but whatever, it doesn't matter. We're using mice today. You're doing mouse experiments, and you have 10 mice, and you want to see what the ID50 is. Whatever dose you give those groups of 10 of mice, whichever dose makes 50% of those mice sick is the infectious dose 50. Whatever dose kills half of those mice is the lethal dose 50. Does that make sense? There's some graphs here too, I'll show you. Pathogens can lose genes, they can acquire genes. We talked about this before. Bacteria are notoriously lazy. If they do not need to be doing something, they do not do it. If you take bacteria, say you have an isolate that is resistant to a bunch of different antibiotics, and you grow it in this lab over and over and over again on like the tryptic soil and plates they gave you all night, eventually it's going to lose those resistance genes because it's not grown and being exposed to antibiotics. So it's just going to get rid of them because it doesn't need to survive. Why is it going to replicate if it doesn't need to? So eventually they become attenuated, so they become weak. That's one of the ways you can make vaccine strains. You just grow them and give them everything they want over and over again until they get rid of it and wimpy. People have done that in the past. There's been a lot of studies who've done it with rabies virus. That's where the rabies vaccine originally came from. Just grow it up over and over again in the animal until it becomes really wimpy and doesn't really do too much. And you use that as a vaccine strain. And again, they can acquire different genes too. So all that to say is the pathogen can change its virulence based on acquiring or losing genes. Same thing somewhat holds true for us, except we're not really acquiring or losing genes, but our immune response can change. So if you're really young or you're elderly, you have a weak immune response. So it changes over time. Maybe you become infected with like mononucleosis or something, and you have a hampered immune response, or somebody has HIV or AIDS, they have a hampered immune response because it's targeting their immune system. So all those things change over time, depending on age and different kinds of infections you can have. One of the first steps of actually causing infection is whatever the microorganism is, it actually needs to somehow attach to your cells. If they don't have the ability to do that, they don't need to stay. They have to attach somehow. So if this is some kind of microorganism we're talking about that's a respiratory tract pathogen, it has to figure out a way to get through that mucus that's overtopping the mucous membrane, get past all the IgA antibodies we talked about, get past things like lysozyme and things like that, and then also express something on its surface that allows it to attach to those cells underlying your mucous membrane, or underlying the mucus on your mucous membrane. So there's a lot of things that need to happen for an infection to actually occur, which the more you kind of learn about this stuff, it's kind of surprising that it ever happens, because it has to get around your immune response, it has to get around your adaptive immune response, your innate immune response. It has to have the right kind of virulence factors. It has to be able to attach. There's lots of things that need to happen for infection to happen. So usually how pathogens are going to attach to you is through things called adhesives. So adhesives allow appearance. The virulence factors are going to be different between different bacteria, like we've been talking about. The way you kind of estimate how virulent a pathogen is, again, is by using different numbers like ID50s and LD50s. Let me ask you that. So if I gave you an example of a pathogen, which one would you be more scared about? One that had a really low ID50 or a very high ID50? Very low, because it doesn't take as much to get you sick, right? Same thing holds true for LD50. Because, I mean, if it doesn't take that many to kill you, that's like rabies virus is one virus particle can cause an infection, and it's 100% mortality. So I guess technically there's not an LD50 with rabies. It only takes one. You can't do the half virus. So there are some nasty examples of this. And then there's some where it takes hundreds of thousands, if not millions, of students. Ebola, ID50s, 10 to 100 viral particles, not a lot, but again, like we talked of rabies. There's 4 examples. Ebola is another one of the ones where the Ebola you see now doesn't seem to have the mortality as much as the original Ebola strains that we saw. Some of those strains, we'll talk about that a little more tonight, like Zaire Ebola, could have as much as 80% mortality. Some of the original kind of Ebola strains we came across were up in the 90s. I mean, some of those were very, very lethal. And again, just remember, like, human host response, the human immune response, the pathogens response to different pressures, all those things, none of them are constant. They change, they fluctuate. Major factors in terms of virulence, these are different microorganisms. Again, the immune response of affecting the individual, how much you were exposed to. So for a lot of these things, the more you were exposed to, the more severe your symptoms are going to be, because you're exposed to more. So there's going to be more there to cause more damage faster. And then how virulent the pathogen is. Is it one that's really not feeling that much, or is it one that's like rabies? It takes one virus worth. Some examples of what I talked about with Ebola there a little bit. Ebola Zaire between 60 to 80% mortality, 40 to 20% depending on what study you look at and what population you looked at, recover. But again, some of those studies and some of those early infections with Ebola were up to 90%, so they can be really nasty infections. And hemorrhagic fever virus. There are some individuals, and they are few and far between that do exist, that are actually to be exposed to Ebola and be asymptomatic. Reason being, I do not remember what the receptor is, but since they don't express the receptor Ebola, yourselves. Those people are few and far between. There are some people like that with HIV and AIDS. They don't make the receptor. What you can do is you can try to exploit that, but what has happened in the past when those tried to be exploited, essentially you give people like your radiated bone marrow and you start to kill off certain populations of cells. You see HIV go away in those people, but then it comes back because there's more than one receptor you can recognize. So there's been a little bit of problems with that, but not as much as the original code. But again, one of the main things for infection to occur is it has to attach to something. If it doesn't attach, it's not going to make you sick. Just like I always give you the influenza example. You can take a bubble bath and a big vial of influenza virus. If you don't get that influenza virus in your respiratory tract, you ain't getting influenza virus. Because that's where the receptors are for influenza virus to attach to, is in the respiratory tract. Nowhere else in the body makes it. Let me show you some graphs. Well, let me actually tell you about that a little bit too. The ID50, so again, you're looking for how many pathogenic particles, virus, bacteria, what have you, make 30% of the group sick. Indicates the ability to infect, but doesn't really say anything about the severity, the rate of transmission. The LD50, by the same token, tells you how many viruses, bacteria, whatever you need to kill 50% of whatever experimental population you're looking at, but it doesn't say anything about how easily it infects. It just tells you how many it kills. It doesn't say anything about how transmissible it is. So these things tell you certain things. How infectious it is, or how many it kills. It doesn't really tell you anything else, really. Okay. So I gave you this example, I'll kind of give you the ID50 example. But this is the LD50 graph. So these aren't too bad to read. So usually they're set up like this. You have a dose down here, and then you have percent death. So anytime you see percent death or mortality, that's obviously going to be the LD50 because it's the lethal dose 50. If you see anything over on this axis that says morbidity, which is sometimes referred to as the sickness rate, or infectivity or something like that, that's going to be talking about your ID50. So you go over here, you want an LD50, okay, where's 50% at? Right here, you go over and see where it intersects your line, and you come straight down. LD50 for this red line, wherever this is 30. LD50 for this blue line is 50. That's it. Not really very hard graphs to read usually. Which one would you be more concerned about, the red line or the blue line? The red line takes a lot less to kill you, right? So they're not too hard to read usually. This is asking me the same thing. I wouldn't give you one on a log scale, but you read it the same way. Well, let me ask you this, which one is the ID 50 and which one is the LD 50? Which one's the ID 50? Left or right? Left, because you look at infection, so infectivity. And over here, you look at mortality, so that's your LD 50. 50%, go over, see where NSX line, drop down. 50%, see where NSX line, drop down. That's how you get the doses. It's not too bad, right? Good. Let's see what else. Okay. So, another question. So, if the ID 50 and the LD 50 are really close to one another, versus an organism where the ID 50 and the LD 50 are far apart, which one would be the scarier one? Close together, because you're sick and you die. There's not much difference in doses between having an infection and succumbing to an infection. So those are the really scary ones. All right, good. We talked about this. Bears are usually used to look at these kind of things, but you can do it in other models as well. Don't worry about any of the math here to do like wellness or riding model, modeling and stuff like that. You do not need to know that. But what I do want to talk about is R0 or R naught. So when we talk about R naughts, this is another way that you try to figure out how microorganisms, how pathogens can spread. So if you're looking at an R naught value, you're trying to calculate how many people one person can affect. That's all R naught is. So the higher the R naught value, it means one person, so say your R naught is 500. It means one person, 500 people sick. If your R naught is 1, it means one person will make one person sick. Low R naught, good. High R naught, very bad, depending on how high it is. So there's some things we worry about when we look at high R naughts are things that aerosolize easily. It's just a little bit of a difference when we talk about droplet transmission versus aerosol transmission. And before COVID, most people haven't heard of this stuff. After COVID, I feel like probably most of you have heard of this. Droplets are bigger particles, so they don't travel as far. So anything that travels through respiratory droplets, like through sneezing, coughing, things like that, a yard, or a meter or less is considered droplet transmission. If it's more than a meter, it's aerosol. It's smaller particles that travel further. Things like measles have higher R naughts. One person may 12 to 18 people sick because it aerosolizes. It can travel further than a meter. Other things have lower R naughts. They don't travel as far, usually because they transmit through droplets. And you see different, you don't need to know any of these, but you should be able to kind of look at this as like seasonal flu has an R naught of 1 to 2, means one person can get 1 or 2 people sick. Measles has an R naught of 12 to 18, which means one person gets 12 to 18 people sick. It aerosolizes better, travels further, gives that one person the ability to make more people sick than just like a regular seasonal flu. So that's kind of how that R naught sounds. And some of these can spread really easily. What you can do with these is you can actually use them to put together a kind of little tree of initial kind of if you have a population of people that are sick, what you can do, depending on where they were at, who they're exposed to, you can start to track it back. And you can, and the first person that had it is the primary case. Whoever that person spread it to, those people are the secondary cases. Whoever they spread it to, they're tertiary, quaternary, and you can put together like a little tree almost that looks like this. You can track back. Sometimes, obviously, the further out you get, the harder it is to track it, but you can start to track back to where the original case was. It's a lot easier to do in a controlled environment like a hospital. You have somebody, I'm not bad jumping down up there, but you have like key card access to things. You have, let's say, like a nurse goes in and interacts with a patient that has MRSA and doesn't change their gloves. They leave that room, they go somewhere else, they card in, and then people behind that door, you see cases of MRSA. And then someplace else. Now, since in a lot of these places we have like key card entry and we can kind of track where people are going and log into different computer stations, into the electronic medical record and things like that, you can be like, well, James was in this room, and then he was in that room, and then he went over there, and you can go back and be like, MRSA, 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 and you can start to tie together. It's just like hey dude, like change the gloves maybe, and don't give the entire board like immersion infection. So you can do that. It takes some finesse, but you can oftentimes do that. I've had colleagues that have done that before. They're like hospital epidemiologists. It's very cool to watch them be able to do that. Or maybe a little scary because you start to realize that pretty much everything you do and everywhere you go, they can track you. But you can start to piece that together. So that's where that comes into play. It also comes into play when we talk about things like herd immunity. So herd immunity is what percentage of the population has to be immune to a disease to protect people from getting an infection that are not immune to that disease. This is where different vaccinations come in. Depending on what the R naught is, some infections are harder to prevent from transmitting from one person to another. So those ones need a higher percentage of the population to be immune to them. So what's that mean? It means if you have something that has a very high R naught, like measles, that spreads easier. So more people need to be immune to that, like through a vaccination, to protect the people that do not have that vaccination. If you're talking about something like seasonal flu that has a lower R naught, that herd immunity, the percentage of people that are immune to it that have had that vaccine, hopefully it's an effective one that year, it can be lower because it's not as transmissible. So where this comes into play is just not just say, it's just not necessarily that you're looking at a pathogen where people have decided like not to go get the vaccine. Sometimes there's people that can't get the vaccine. There's different kinds of seizure disorders and things that if people spike a fever, I can never remember the name of this disease, but if you spike a fever, whether it be from a vaccine or actually an infection or for any reason, they have seizures. Yeah. So there's people that have that kind of condition and other kinds of conditions or religious exemptions or whatever, they can't get a vaccine. So if you don't have everybody else essentially immune to it, it makes it a lot easier for them to get infected. So I'll give you an example of religious exemption. So there was a Orthodox, is in New York, I'm pretty sure. There was an apartment building and a lot of Orthodox Jewish people live in that building. They don't believe in vaccines for measles. Measles spreads really easily. So what happened is somebody got measles, they went in, got an elevator, touched the button, and literally everyone that used that elevator, the entire building got measles. So that's kind of a worst case example because you had a collection of people that none of them were immune to it. But out in the population where you have some that are immune, some that aren't immune, the chances of somebody that's not immune coming into contact with somebody else that's not immune that has the infection is really, really low. Unless that's risky.
When you talk about like, okay, there's like someone's in an apartment building and someone from the kids and they have measles, is that within like a specific hour time? Like so like within like 2 hours, 3 hours?
So that's a good question. That depends on the pathogen too. So some pathogens survive better out in the environment for a longer period of time than other pathogens. With that example, I can't remember how long measles survives in the environment, but it didn't really matter because you have one person come home from work 15 minutes later, somebody else. It was so fast that they were just all exposed to it over and over and over again. I mean, even, I kind of just went past this. This is another example that's a little bit more graphic, I guess. But like old school SARS, like SARS-CoV-1 back in 2002, they tracked a pandemic in Singapore where essentially what people would do is like one person had it, they would flush the toilet and the stuff would aerosolize from them through the sewage pipes and stuff. And the whole building got it pretty much.
Fun fact, that actually does happen when you flush the toilet or the guy's pee standing up, it gets everywhere. So.
This is another one that if you ever go on a cruise ship, norovirus spreads super easy. I can't remember what the R naught is for it, but it's high. And it aerosolizes, and what happens is if somebody goes to the bathroom, it can aerosolize. What can also happen is sometimes it causes such a guttural like vomiting that the people, when they vomit, particles aerosolize when they vomit, and that can be carried to people around them. Like this is why if you're in a confined environment, like on a cruise ship, and one person has norovirus, that whole cruise ship's gonna have norovirus, that's what, it's bad. Yeah.
So for like healthcare workers, how protective is PPE against some of these viruses?
N95 mask, filter out 95% of airborne particles, so N95s work really well, which is what people should have been using when COVID-19 was raging. The problem with that is we weren't prepared for that. We ran out of N95 mask, and then suddenly toilet paper became an acceptable replacement. So, I think what that was is we didn't have a stockpile, so I think they just wanted to make people feel good, I guess. I mean,
So
something.
those like the regular masks.
Right. I mean, something's better than nothing, but I mean, does that protect you anywhere near as much as like a N95 mask? I mean, that's not all. But I mean, if you don't have a N95 mask, then, I mean, you wear a N95 mask, you can wear that in BSL-3 lab. I mean, that's what we always wore, unless we have like a PAPR or something, but I mean, that's fine. You can work with things that aerosolize easily, that have low infectious doses with an N95.
5% risk the entire time.
Yeah, but like in theory it's 5%, in practice it's less. Like if you're wearing a N95 mask with anthrax, you're not gonna get anthrax. You have to have some kind of a breach usually. It's just like somebody has a 95 mask on and they're like, and they take it off. It's like, okay, well now, yeah. Like if you breach that barrier, yeah. And this is why they do fittings and stuff, like if you're working around a hospital and you're doing anything like that, usually they have like a fit test where they make sure that it fits your face correctly and things like that, just to make sure you don't have a breach. Because if you're wearing your N95 mask and you have like, you can stick your fingers behind it like this and there's holes, like obviously it's not fit to your face well. This is why they also kind of watch, like if you have a lot of facial hair, it doesn't fit as well because the hair gets in the way. So I mean, if it fits correctly, you should. But yeah, some of the masks got pretty thin towards the end of that. I remember picking one up and it was disintegrated in my hand. It's like, well, I don't think that's probably gonna help. This kind of puts it in graphical form of what I'm talking about. So over here you have, so red means they're infected, blue means they're not infected. Or sorry, red means that they're not immune, blue means that they are immune. My bad. So this, you have one case, they're not immune, none of these other people are immune, everybody gets it. Down here, you have one person that is not immune, so they haven't been vaccinated. They're exposed to these people, one of which is also not immune. That non-immune person can spread it to other people, but it essentially stops with all these people that do have immunity. So it's just less, the more people that are immune against some of these different pathogens, the less likely they can spread it, because there are more people who are immune to it, that's all. That herd immunity, what the percentage is to actually have protect the entire population changes depending again on what the R0 is. Ones that have higher R0 that spread easier, you have to have a higher herd immunity, so a higher percentage of the population immune. Ones with lower R0, you don't have to have that high. Usually a lot of those things, the herd immunity range from 85% and above is usually the kind of range that we're looking for generally. Morbidity rates versus mortality rates. So morbidity rates, again, sometimes referred to as sickness rates. Morbidity rates are the number of new cases of disease during a specified period of time over the total population of individuals that are susceptible to that infection. So what does that mean? It means if you're looking at morbidity rate for syphilis, you look at the number of new cases of syphilis, which is technically the incidence, when you're looking for new cases. Look for the incidence of syphilis over how many people were exposed to syphilis. I mean, if you're looking at like a sexually transmitted infection, when you calculate these rates, it has to be in the population of people that are susceptible to it. So if you're looking for sexually transmitted infection, and you're looking at the population, and you're including people that aren't sexually active, then that's not the right rate. Because you're including people that are susceptible to the illness. So that's sickness rates. Mortality rates, what it sounds like, number of people that die or the number of people in the population that have that disease. So again, you're looking at the people, you don't want to take mortality rates from the entire population if you just look at one disease process, because you can count a bunch of people that don't have the disease. So you look at how many people died from the group of individuals that had that disease, that's your mortality rate. So it's just some kind of definition stuff. Again, how are these things transmitted? We talked a lot about this. You can have droplet transmission. Another little kind of nuance with the terminology here. If droplets are spread, or aerosols are spread for a meter or less, it is considered direct contact transmission, even though you're not necessarily touching the person. But think about what that means. It means if you're this close to me and I sneeze, I've sneezed in your face. I didn't shake your hand or give you a hug, but I sprayed saliva into your face. So it's not, I understand it's not direct contact, but it really is considered direct contact. If it's anything more than a meter, it's considered indirect. Does that make sense? That's kind of nuanced with the terminology there a little bit sometimes. So what's an example of indirect transmission? I can't remember for the life of me where this experiment was done now, but it was on the East Coast, I want to say. There was a professor who was doing this kind of research, and he took Serratia, we haven't worked with Serratia in here, have we? Serratia marcescens sometimes makes this really kind of pink pigment. Usually if it causes an infection, it causes a urinary tract infection, it doesn't do much else. It can be a pathogen, but it doesn't do a lot of stuff. What he did is he took a strain of this that was non-virulent, and he swabbed it inside his mouth, and then he gave a lecture. On the back of the room, he had a bunch of petri dishes that were open. And as he talked, what he started to figure out, he went back and plates after the lecture, put them in an incubator, and he had all these pink colonies all over them. Because just him talking, he made aerosols from talking that goes all the way to the back of the room. So just regular kind of talking spreads things when you're in a confined environment. And this was not a lab like this. This was like a 300 student like lecture hall. This was like a big room. Yeah. Reason we have IRBs and things for lots of things. Yeah. No, I don't believe it was empty. I think he just delivered classes with about 300 students. There's more controls on our experiments now. They calibrated a lot of things before. Anything before the 90s was kind of like the Wild West. Things got a little bit better after the 90s, at least for that stuff. But yeah, I mean, it does transmit a lot of things pretty easily. I mean, truth be told, I mean, the federal government has done these experiments too. I mean, they've taken, because I understand how some of these things spread, in order to do defensive, don't ask me how I know this, to do defensive bioweapons research, you have to understand how some of these things are spread, so you essentially are designing bioweapons. So you take a flight, you want to see how anthrax spreads. You take a C-130 and you put a strain of anthrax that's either a variant in it, or you put spores from some other bacillus species that's not toxic to people, like an environmental sample, like, I don't know, Pathogenicity or something, whatever, something that doesn't make us sick. Make a bunch of endospores, you pack in a C-130, and you fly over a couple states and you drop it out. And then you have collection sites where you go down, essentially like that professor did, it's just like, well, which place do I get Pathogenicity from? And those things have been done. It doesn't make anybody sick, but I mean, in order to try to figure out what to do, like if somebody did something like nefarious, you have to know how that would spread. So I mean, U.S. AMRID, U.S. military, like Army Research, infectious disease, they've done these experiments. I know that because they told me that. And those guys are really scary if you ever want to be like horrified and not sleep for a few nights. Yeah, they've done some things. But you have to understand how that stuff works to be able to come back if somebody does something not so great. Which is why we have warehouses and warehouses and blocks. What else? Vectors we talked about. So vectors are usually talking about arthropods. Fleas, ticks, insects, things that The pathogen, as part of its life cycle, has to go through those vectors, and then those vectors again bite us. We're usually an incidental or an accidental host. We have nothing to do with their life cycle, but somewhere along the way we get transmitted to us. We get sick. Fomites, those are anything that are inanimate objects. So doorknob, computer, phone, desk, the lab bench knob. Somebody comes in, they sneeze on the door handle. You touch the door handle and transmit the infection to yourself that way. The door handle's a fomite. Anything that's inanimate that can harbor pathogen. Excuse me. Parental contact is when you get a needle stick and somehow get cut. So if somebody comes in, you're seeing a patient. This, I think, that's how you know this. My wife had like a cut on her finger, like right as soon as we got married. So somebody came in, this was in Cincinnati, somebody came in, traumatic injury, they were in a car accident, and I think one of the younger residents was trying to cut off their clothes, like just decently cut through my wife's ring finger. So like, yeah, this, I hate wearing my wedding band, I don't know why. It's like, I hate wearing that. So like the ring was here and it missed a ring by like a millimeter. Like if it just would have been like just the hair the other direction would have said a wedding ring. But it cut through her finger. It didn't like take the finger off, but nice healthy cut in the finger. Well, you don't know, I mean, traumatic car accident, you don't know if that person has hepatitis, you don't know if they have HIV, you don't know what they have. So that was our first week of marriage, I think. So you're doing like HIV prophylaxis, hepatitis prophylaxis. It was fun. Yeah. So that's parental. So you're transmitting through some kind of a sharps injury. I teach my ID class about this a little bit. So sometimes people get like horrified. This was way off topic, but sometimes people will be scared they're going to get in trouble when something like that happens. It happens every once in a while. Sometimes people get a needle stick or cut with something. You have to go tell people, like the infectious control nurse usually is what they call the people at the hospitals. You have to tell them. One, because the hospital gets in a ton of trouble if things aren't reported. And two, you want to make sure that they test who that patient was for all these different things before they leave, because sometimes people will abscond and then you don't know. And that's not a fun way to live. So they usually take a couple blood samples and see if they have anything that's transmissible or not. And then lastly, you have vehicles. So vehicles are like the peanut butter example in the salmonella I gave you. It's food or water or drink or what have you. There's something in it, you consume it, that's a vehicle. Salmonella doesn't need peanut butter to live, obviously, but you can eat the peanut butter that has salmonella in it. Salmonella would be, or peanut butter would be a vehicle. If you're talking about water, the easy example to give of this is cholera. Somebody goes to a drinking well, there's fecal contamination of drinking water, they consume some of the water and they get cholera. So water would be a vehicle. We talked about this. Highly communicable diseases are usually transmitted through droplet transmission or mist aerosolization. The ones that are highly communicable are usually spread through aerosols because aerosols travel further before they drop out of the air column. As a particle travels, that droplet travels further and further away from you, say you sneeze or something, the water evaporates in that droplet, the droplet gets smaller and smaller, and sometimes things can be carried that further depending on what the pathogen is. Some pathogens carry further than others. Some examples of this, similar examples. We talked about the SARS-CoV-1 example here a little bit ago, that's at the top of this slide. Another example I haven't talked about yet is Neisseria meningitis, actually in northern Africa. What can happen is when it's super dry, not when it's not rainy season, that can be carried in the air, and a bunch of people can get meningitis. That's why that's called meningitis belt. So every time that it's like this time of year during dry season in North Africa, we expect a certain number of meningitis infections. That's endemic. If it increases above that threshold, then it would be an epidemic in that area. About 200,000 individuals infected annually, 25,000 people die, so not an insignificant number. But again, it's something that in that area it is endemic. different microorganisms can exist in an environment for different periods of time. Some can survive desiccation, so it's drying out for a longer period of time than others. Some things can survive on different surfaces better than others. Some microorganisms, like some bacteria, can adhere to glass better than plastic, or Teflon better than glass, or what have you. It depends on what adhesive molecules they have on their surface. Different things can adhere to different things easier. Different portals of entry, so how the pathogens actually get into your body. They can go through mucous membranes, and some major portal of entry, like your respiratory tract, your eyes, and your gastrointestinal tract. They can get into the cracks, like I talked about with the Purell, the old school Purell a little bit, cracks in skin, get through hair follicles, get in through sweat glands, or they can get in orally. So something from the soil, there's a lot of different microorganisms that live in the soil, obviously. You consume, say, produce that has a little bit of soil left on it, and on that soil there's some bacteria that make you sick, so that would be soil to oil transmission. Fecal to oil transmission, I don't think I need to explain, but that is what it sounds like. Yeah, let's explain that anyways. I always give the Chipotle example. I feel like I gave them a bad rap over the past 2 decades. You go to Chipotle, and somebody goes to the bathroom that's making your burrito, and they don't wash their hands, and they come back out, and they finish making your burrito, and they were carrying hepatitis A, and you haven't been vaccinated against hepatitis A. You now have a burrito and hepatitis A. So, I mean, that happens in Greenville. Whatever. I don't know for the health department, what are they going to do, fire me? So in Greenville, what happened was, for whatever reason, there was a lot of different restaurants that had people working in the kitchen that had hepatitis A. So you saw a lot of people that were coming into the hospital with hepatitis A, and it's just like, this is not like an endemic, like, oh, like 3 people had hepatitis A. This is like 50 people had hepatitis A. Like, this was like getting to like an epidemic kind of deal. But then exactly at the same time, COVID-19 hit. So the health department pretty much were like, we got our hands full on this one, and they stopped monitoring. So you had COVID-19, and don't go eat anywhere, not that you could anyways, because there's hepatitis A in all the restaurants. So we watched those things unless there's a pandemic, and then kind of the rules get thrown out the window. But that exists in a lot of places. I think most people, you don't know when you've been vaccinated against for hepatitis. You've been vaccinated for hepatitis A and B, I'm assuming, correct?
Yeah.
So back in my day, the hepatitis A, hepatitis B vaccine was not a thing. It was the combo vaccine. They gave you a hepatitis B vaccine. So I was vaccinated for hepatitis B, so I went back and got hepatitis A. The vaccine, not. But yeah, so I think most people probably have had the combo vaccine now. So that's usually a short-lived illness. You have diarrhea, you can get some jaundice associated with those hepatitis viruses. Very unpleasant. Microbes can enter the human system during dental procedures or trauma. I don't think talked about this either. There are some nasty microorganisms that can live in your mouth associated with dental plaque. If you had come across something that has periodontal disease, some of those bacteria can actually escape the oral cavity and migrate primarily through tissue to other areas in your body that are close. It's called a focal infection. If it goes to another area of the body that's close by, primarily through tissues, usually ends up going to the heart and causing endocarditis. I think I talked about this in here with porphyromonas, gingivalis. That's something you find inside your oral cavity. They found proteins from that called gingivanes in people's brains that have Alzheimer's disease. There's some correlations to things in your mouth getting other places and causing nasty things. What even can happen sometimes is you can get something referred to as transient bacteremia. Everybody talked about what bacteremia is in here yet? Anytime you see emia, it means you got something in the bloodstream that's not supposed to be there. It's like a microorganism. Bacteremia, you got bacteria in the bloodstream. Fungemia, fungus in the bloodstream. Parasitemia, some kind of parasite, like say malaria or something. Viremia, you got viruses in the, so just, you put whatever is in the bloodstream in front of emia is all it is. You can have something called transient bacteria. If you have somebody that brushes their teeth really hard, and it gets to the point where like there's a little bit of blood, well, some of that stuff can get into your bloodstream, but it's transient, so it doesn't last long because your immune system typically wipes that out. If, the other example is more graphic, if you have somebody that had a very strenuous bowel movement, they can sometimes get normal flora into their bloodstream, like say somebody was very constipated, and they could have a transient bacteria from that. So sometimes you have a little bit of bacteria in the bloodstream, but usually again your immune system wipes it out because it's a low dose, it just takes care of it. But again, if something gets from one area of your body where it's supposed to be to another area of your body where it's not supposed to be, it can cause different things to happen. Coronary artery disease, chronic inflammation of the joints, implants, arterial walls. So things can go to places they're not supposed to be. Form biofilms are really hard to get rid of. Again, for infections that occur, there's a lot of things that need to happen. First and primarily, what needs to happen is some kind of an attachment. After that attachment or adherence to your cells, it's going to start to colonize the tissue, generate a localized point of infection in the tissue. It might start to invade the tissue, so it can just start to grow so much that it actually causes some kind of an inflammation. A good example of this to give is how different kinds of meningitis, bacterial meningitis, can start. Things like Neisseria meningitidis and Haemophilus influenzae and strep pneumo. They can move to your blood-brain barrier, cause inflammation in those cells, and then once they cause enough inflammation, those bacteria can start to find their way into your cerebral spinal fluid and cause meningitis. So that can be due to overgrowth of the blood-brain barrier, or migration into additional layers of tissue. Some of these microorganisms make different virulence factors that allow them to get through your cells. They might make a collagenase. Collagenase breaks down collagen. They might make a hyaluronidase. Your cells make hyaluronic acid on the outside of them. Hyaluronidase breaks that down, so they can find their way through those cells and making different virulence factors. Or they might just straight up slice the cells. You might make lipases, phospholipases. Continue migration to the tissue allows the microorganism to penetrate into the bloodstream. Once it gets into the bloodstream, it can cause a systemic infection. And depending on what the pathogen is causing that, if it's something like a gram-negative, remember gram-negative cells have lipopolysaccharide, and a component of lipopolysaccharides is lipid A. Lipid A is endotoxin. So endotoxin can cause septic shock. Depending on what's in the bloodstream, it can cause some pretty nasty side effects like a cytokine storm. Different ways these pathogens can attach. It can make capsules for slime layers. Strep mutans does that. It actually breaks down sucrose to actually make the capsule, and that allows it to attach to your teeth and your mouth, obviously. Oh, I had to justify that one. Adherence proteins or polymers. Techoic acid and gram-positives can allow the gram-positives to attach to your cells. Influenza binds specific receptors on the epithelial cells in your respiratory tract. Different microorganisms can make fimbriae or pili for attachment. E. coli 15787 makes fimbriae that attach to enterocytes. Enterocytes are the cells that line your intestinal tract. That's what allows E. coli 15787 to cause hemorrhagic infection. Then you get the E. coli example here in your urinary tract. So E. coli, some E. coli, you can have E. coli in your urinary tract. If it doesn't actually make the villi that attach to your epithelial cells, it doesn't cause urinary tract infection. So it has to adhere somehow. If it doesn't have the villi to adhere, it can't cause UTI. So it just has to find a way to attach. Strep pneumonia, which can have a capsule. A capsule is how they attach to our cells, invades. You get phagocytic cells, and then the phagocytic cells are either unable to phagocytose them, so to take them up, or unable to actually digest them once they actually do phagocytose them. There's other ways your immune system uses to get rid of those encapsulated bacteria. Usually what they do is they coat them in antibody and activate components of the complement system. Remember we talked about complement? How do we talk about complement killing cells? All the little pieces and parts come together to make a what? Make a what? A what? Yeah, membrane attack complex, they make a pore, those holes. So encapsulated, or so in, and as a membrane, including viruses that have a envelope. What happens sometimes is you have somebody that's asplenic or functionally asplenic, so say that they had some kind of a tearing car accident, they lost their spleen, or they have like sickle cell anemia, red blood cells are went into the small vessels in the spleen and actually started to tear up the spleen, so the spleen's there, but it's not functional, so they're functionally asplenic. And that's where a lot of these things happen with trying to clear bacteria that have capsules. So you have to make sure that pretty much these people that are functionally asplenic or asplenic have all kinds of vaccines against pretty much anything that has a capsule. Because your spleen's usually where those organisms are here. So things like strep pneumonia. The colonization depends on a bunch of different things, availability of nutrients, so there's not a lot of nutrients there, bacteria's not going to thrive, the optimal growth conditions, such as temperature. Did I show you all the necrotizing fasciitis case in here yet? I probably forgot everything. You think might be potentially possible infection. And then after about 48 hours, you do something called an antibiotic timeout. It means you discontinue antibiotics, you go look at lab results, see what it actually is, cause an infection, see what it sends, what your antibiotic is sensitive to, and then switch it from that broad spectrum antibiotic to something that's more targeted. So it's a human targeted therapy. The problem with this is by the time they get the lab results back, or
So because the lab results
well, I mean, there's nothing, I mean, you gotta throw it up. The problem with this is, I'm sorry. Problem with this is it wouldn't have mattered anyways because it was resistant to everything we had. The only thing that it was sensitive to is an antibiotic called colistin, which you typically don't use for this. This is just like colistin. If you're using colistin, you can't even pull colistin out of the formulary usually in hospitals without like having somebody from a pharmacy cut that and sign off on it. They do not want you to use that because they do not want things to become resistant to it because we don't have anything else. Like if you're at a point where you're using colistin, you are out of options. You were done. And that was the only thing those were sensitive to. A year later, somebody else came out with similar symptoms. Very similar microorganism, very similar antibiotic resistance profile, and then it disappeared and never saw it in the hospital system again. It was the,
Did
I don't
person
know.
that year later also die?
within about 72 hours.
Where is this found?
Acinetobacter?
Like the bacteria? Water?
We don't know necessarily. Acinetobacter has some genes that look like it's a soil bacterium. It has some genes that have some relationships to some different things made by bacteria you find associated with plant roots. So maybe once upon a time it was something to do with like plant rhizosome or something. We don't know. It's a soil bacteria, it's usually what we say. Worst case scenario, obviously, these kind of infections you see with people that are very immunocompromised. You don't see people that have normal immune response to these type of infections. These people both were very, very, very immunosuppressed to the point where, to be real honest, they didn't have much of an immunological response at all. I mean, their immune system was just off, essentially. So there's not much they could really do about it. Those pathogens in particular, at other places, I have done research on those. We think they spread so fast because they make, I think I talked about this, they make this really long pellets. And at the end of the pellets, they have a tip adhesion, and they use it to take the bacteria and pull it towards that tip adhesion. So the pellets are just like ginormous, so we think that like let it move really fast. One of my colleagues I still collaborate with does some research on these strains too. And we also have been starting to figure out that they actually live inside macrophages too, which is something else we haven't known about. But somehow they figure out a way to go intracellular and hang out there. So it's interesting. Yeah, sorry.
Yeah. I just have a question, like, do you think the process of the bacteria
Yeah, so I can't remember everything they were on. Yeah, they were on corticosteroids. I remember they were on rituximab, which anytime you see a drug that ends in MAP, means monoclonal antibody. They're on rituximab for lupus and TTP, which is another immune disease. Yeah, again. And I think the second patient had AIDS, and something else. They had multiple immunological conditions that made them very susceptible to this. And to be real honest, sometimes those patients that really don't have much immune response at all, if that bacterium wouldn't have done it, something else probably would have. Because if you can't fight it off at all. Yeah. But, wow, big tangent, sorry. So that, the temperature plays a role in this. The growth factors, so remember some of these microorganisms are really picky with the things they need. If they don't have the vitamins they need to grow, they're not going to grow. If they don't have iron, they might not be able to grow, especially if it's a microorganism that can't scavenge for iron. To say that they can't, make things like siderophores that can take iron away from different proteins in the body. Some bacteria can make siderophores, some bacteria can't. One of the ways that your body actually kind of suppresses the growth of a lot of these pathogens is it sequesters iron from them. They don't have iron, they can't grow. Different kinds of infections. You can have a localized infection, which is like a boil, localized in a certain region. You can have septicemia, which means it's everywhere, it's in the blood. And then you can have different microorganisms that make toxins. Clostridium tetani is an example of this. Clostridium tetani makes tetanus. It's causing infection. It can secrete that toxin. It can cause tetanus. Tetanus causes what's referred to as a generalized spastic paralysis. It means all your muscles contract. As opposed to botulism toxin. So usually, I swear I put this up here somewhere. Botulism causes a descending classic paralysis, so your muscles relax. So botulism, I think I talked about this in here before. You worry about as the effect descends, it causes the diaphragm to not be able to contract anymore, so people can't breathe in, so you have to put them on a ventilator. Invasiveness, ability of pathogen to cross the host tissue and clear function, pneumonia spreads across lung tissue, also the ability of pathogen to invade into tissue. Different examples here, they can continue to migrate through the tissue, and that's because they make different virulence factors. Expressions of the different virulence factors is energy expensive. So what that means is it takes a lot of energy of these bacterial cells to make these different virulence factors, so they don't make them unless they need to, because they're lazy. The virulence factors usually are not expressed until a certain cell density is reached. Usually that cell density is around 10 to the 6 bacterial cells, and at that point, the bacteria are considered to have reached a quorum. And they determine how many bacterial cells are surrounding them by they each secrete quorum sensing molecules. And when a certain number of quorum sensing molecules are reached, the bacterial population know that they have reached a critical white cell mass. And then they can start making different virulence factors. I'll have a good example for this here in a moment. I'll make a little more sense of it. So this gives the pathogens a head start with the immune system. Go to the next. Oh, here we go. So, where does that come into play? So, different isolates of streptococci can make these two virulence factors. They're actually enzymes. They can make a coagulase and they can make a streptokinase. So early on in infection, they make a coagulase. Makes fibrin clots so the pathogen hides in the host fibrin. So it robs fibrin from your body and shields itself with your fibrin so your immune system doesn't see it. Once a critical amount of bacterial cells of streptococci exist, they've grown up, they will stop making the coagulase and they'll start making the next one down called streptokinase. Streptokinase breaks down the fibrin so they can spread somewhere else. Would they make the coagulase and streptokinase at the same time? No, because if you're breaking down the fibrin, they're trying to hide out in it. So that expression of those different virulence factors is controlled by the amount of bacterial cells there. Makes coagulase first to hide itself. Once there's enough bacteria there, it's not going to matter if you have an immunological response or not, because you have so many streptococci cells that are going to overwhelm the immune response. So they start making the streptokinase to break down the fiber and spread. Yeah.
So it's when they start making streptokinase, that's when you start kind of getting like these symptoms, because that's when your like immune system starts kind of recognizing it.
Potentially. Yeah, you'd be more symptomatic at that point. Technically, it's considered the disease process if there's damage even to one cell, so it's ripping fibers away from a bunch of your cells. I mean, you would definitely be symptomatic once it starts making streptokinase, you would expect, but sometimes you might be a little symptomatic even as it's making coagulase before it reaches form. Like there's like a kind of window in there where you start to be symptomatic. It just all depends on how many cells are damaged. You go back. How different environmental factors can control the virulence. Crinobacterium diphtheriae, that diphtheria toxin that causes diphtheria is actually encoded by a phage, and that expression of that toxin gene is regulated by iron. Bordetella pertussis causes whooping cough. Expression of the virulence gene increases as elevated body temperature goes up, so if you estimate the fever itself actually causes pertussis to make virulence genes and cause whooping cough. Vibrio cholerae is controlled. Expression of the cholera toxin genes is controlled by a bunch of things: pH, temperature, salt concentration. Essentially, all things that it can detect in your body that will cause it to start making cholera toxin. Because when Vibrio cholerae is in an environment like, say, in a well or something, typically it does not make cholera toxin. However, when it is in your body at body pH, body temperature, and body salt concentration, it makes cholera toxin. Because why does it need to make cholera toxin when it's hanging out in a well? What's it making a toxin against? There's nothing there except water. Again, they're super duper lazy, so it's not going to make cholera toxin unless it's making cholera toxin to do something. So we talked about a couple of these already. Collagenase breaks down collagen. Lipases break down lipids, phospholipids, and in your membranes. Proteases degrade proteins. What's an example of that? You have things that are going to be able to colonize like your underlying mucous membranes. Like say you have like a respiratory tract infection. Well, what's on your mucous membranes? IgA. IgA is a glycoprotein. So a lot of those things that are going to be able to get through your mucous membranes are going to make proteases to break down IgA to get past them. Nucleases break down DNA, break down things like DNA. Why does that matter? That's kind of an oddball because things like neutrophils can release DNA that have things up in that net of DNA in those neutrophil traps. And these bacteria that make like DNases aren't trapped in those extracellular traps with the neutrophils because those flip up the DNA. A lot of these virulence genes can be transferred between bacteria on these big cassettes of DNA called pathogenicity islands. A lot of times they can be transferred between similar species, but they don't necessarily have to be. They can be more promiscuous and be transferred between different kinds of genera as well. Difference between an intoxication infection and an intoxication and a toxico-infection. Intoxications like the Clostridium botulinum example that we were starting to talk about. You usually have somebody that's home canning foods. They can the food. There gets some Clostridium botulinum in the jar or in the can or whatever. It starts to grow, starts to make toxin. Later on, you open up the jar. The bacteria is probably long gone, but the toxin is still there. So when you get botulism, you're getting an intoxication illness. You're not getting an infection from botulism, for Clostridium botulinum, you're just ingesting the toxin. So that's called intoxication. If you have an infection, and the bacteria that's causing that infection is making a toxin, you have a toxico-infection. So a toxico-infection, you have bacteria actually growing and making a toxin. Intoxication, you just consume toxin. Does that make sense? Okay. And like we talked about anemia, if you have a toxin circulating in the blood, it's called toxemia. Talked about this a little bit. Remember, gram-negatives make lipopolysaccharide. Lipopolysaccharide has that lipid A component that's endotoxin. It can cause septic shock. That's only gram-negatives. And gram-negatives and gram-positives can both secrete different toxins. They're just called exotoxins because they're secreted extracellular of the cells. LPS is considered an endotoxin because it's the component of the cell wall, cell membrane. Some different kinds of exotoxin examples are secreted. Let me hop past AV toxins, I'll come back to them. You have neurotoxins. Neurotoxins target nerve tissue, obviously. Botulinum toxin versus tetanus toxin. So we kind of talked about both of those and how those work. Enterotoxins are going to target your intestinal cells. Staphylococcus aureus can generate enterotoxins. What happens a lot of times with these intoxication illnesses, like say, if you have an intoxication illness versus a toxicode infection, usually the symptoms of the intoxication happen within hours. The toxicode infection symptoms usually happen within days. So the reason for that is, if you open that can of whatever, green beans, seems to always be green beans for some reason, open that can of green beans, and there's botulism toxin in there, and you consume the green beans, you've already gotten your big dose of botulism toxin. The Clostridium botulinum's already grown up in there with those green beans. It's made a bunch of toxin, you are consuming that now. If you have a toxicode infection with a bacterium, that bacteria has to grow up, and up, and it's making toxin as it grows. It takes longer for the bacteria to grow for you to get an amount of toxin in your body to actually cause the effects. So the effects of intoxication illness tend to be within hours. An example of this is like Staphylococcus aureus. Somehow it usually is associated with things that have milk in it for whatever reason. You see somebody go in to like a picnic or when it's hot, they have like potato salad. I don't know why it's always potato salad. They have potato salad that's like sitting out. The Staphylococcus is grown up in potato salad. They consume the potato salad. That Staph aureus is also released as enterotoxin. Within about 6 hours, up to as long as 24 hours, they are very sick. And then they get better over the course of a day or so. They usually just have to be hydrated. But intoxication illnesses move pretty fast, and they're short-lived usually too. Cytotoxins are going to kill cells and lyse them for nutrients. Cytotoxins like hemolysin. Hemolysin are called hemolysin because they lyse red blood cells. Staph aureus can make those too. And then also you can have these AB toxins. So AB toxins, anytime you see a toxin called an AB toxin, it means it usually has a couple different components, an A component and a B component. The B component is for binding. That's the motif of the toxin that allows it to bind onto your cells. And the A component of the toxin is the active component. That's what causes whatever symptom of the toxin we're talking about. So it has an A and B. B is for binding, A is for activity. Different things you see with LPS for endotoxin, fever, shock, blood coagulation, weakness, diarrhea, inflammation. That's on average the symptoms of shock. Leads to a bunch of chemokines and cytokines being released, massive B cell responses that are specific to too much, and it can result in death from septic shock. So those are for gram negatives because of the endotoxin. All right.